Welcome to Georgia Tech. I'm Professor Wilson, and it is my pleasure to introduce the Wilson Lab. I joined Georgia Tech in 2016. Prior to that, I was a professor at Yale University in the Department of Chemical Engineering. And I was recently elected to the American Institute for Medical and Biological Engineers for developing the field of biomolecular systems engineering. And I'll describe what that means or what that is in this video. Before we get started, here's a table of contents of what I plan to cover in this talk, starting with an overview of the research program. I'll transition into a summary of our research accomplishments, followed by a brief description of projects for incoming students, and then I'll conclude with a virtual lab tour. So what is biomolecular systems engineering? Well, this is an NSF-sponsored program in which we're interested in converging the areas of biological engineering, chemical engineering, and electrical engineering. And many of you know that biological engineering is a very large area, and our focus is on iterating between protein engineering, genetic engineering, and metabolic engineering, with the latter two fields representing the discipline of synthetic biology. What makes us a bit different from other synthetic biology groups is our ability to engineer new proteins, which allows us to innovate at a different level. So with this program and with this focus, we're applying it toward the development of intelligent biological systems. And by our definition, an intelligent biological system has three tenets. The first tenet being decision-making. That decision-making can be coupled to a memory structure. And for advanced systems, we can imbue them with communication, either unidirectionally or bidirectionally. The focus of this talk will be on decision-making technology, and we'll work through how we accomplish that through a set of workflows. And we can apply that similarly to the development of memory and communication. So the technology in question for decision-making is transcriptional programming. Now, this is an innovation of the Wilson Lab, and we published many articles in this area. So if you'd like, you can take your time and take a look at them. However, what I'll point out is that this slide is decidedly not very biological in many of the vignettes. And that's because, as I mentioned, we draw our inspiration from other engineering disciplines, specifically electrical engineering and chemical engineering. So in order for you to understand transcriptional programming, you have to know something about transcription factors. So there are many transcription factors that exist in nature. The variety that we're interested in are allosteric transcription factors of the sort that you see in the blue box. So the way this protein functions is that it binds to a DNA element called an operator. That prevents RNA polymerase from transcribing the downstream output. And in the presence of an input, specifically this dark gray hexagon, the protein undergoes a conformational change. It releases operator DNA. Now the system can produce an output. In this case, we're using green fluorescent protein as an exemplar, but it could be anything, including a non-coding RNA. If we digitize the input, we can define the system in the context of a logical operation, specifically an open loop control system in which zero input corresponds to zero output. And only in the condition in which we supply one input under saturated conditions do we get an output. Now, our design goal, at least at the outset a number of years ago, was to create a broad collection of these transcription factors that can respond to a broad variety of signals. To accomplish this, we developed a process called modular design. And this is similar to us walking over to a parking lot full of cars, taking them apart, making a pile of engines, making a pile of transmissions, and making a pile of tires, and mixing and matching those parts so that we can create new vehicles. Clearly, we're not talking about automotive parts. We're talking about parts of a transcription factor. And principally, there are two parts that we're interested in, namely the regulatory core domain. That's the top that processes the input signal. And then the bottom, this is the DNA binding domain, or the engineered component, which we refer to as an alternate DNA recognition unit, which interacts with the operator DNA element. So a number of years ago, we developed our first design space, principally constructed of 6 regulatory core domain and 7 alternate DNA binding functions. And when paired, this creates a putative design space of 30 non-synonymous transcription factors. So we designed, we built, and tested all 35 transcription factors, and to our delight, the vast majority of this design space was functional, validating our design strategy. Noting that this is just the tip of the iceberg, and that our broader design space is on the order of 10 to the 7. So there's plenty of work to do. Next, we engineered the antithetical logical operation to the repressor, i.e. the anti-repressor. Now, this is an innovation of the Wilson Lab, and it's not represented in nature. So this is really a unique opportunity for us in that now we have a powerful tool to complement the repressor. Principally, the way this system works is that in the absence of the input signal, now the system is on, and only in the presence of the input does the transcription factor bind to operator DNA and turns off the output. Digitizing the system again, akin to how we manage the repressor, this corresponds to a single input, single output open loop control system that can be defined as a NOT operation. Namely, 0 input corresponds to 1 output, and 1 input corresponds to 0 output. Now, to engineer systems of this sort, we had to depart from the modular design, at least at the outset, and you have to know something about allosteric communication. Specifically, we had to know how to block allostery at the atomic level. Namely, by introducing a point mutation, we can disconnect communication between the ligand binding site and the DNA binding site while retaining the DNA binding function. And then we can apply directed evolution. In this case, we used error-prone PCR to facilitate the introduction of compensatory mutations and then discover new phenotypes using high-throughput screening. Namely, we were looking for anti-repressors. So initially, we applied this to several structures. And what you can see is that it's generalizable. And all of these unit operations can be complemented to the repressors. And we can apply modular design. So now we have transcription factors that are network capable. To date, we've created more than 100 of these allosteric transcription factors that can interact with a range of inputs from IPTG to caffeine. And we can engineer many components of the system, including the ligand binding site, to create new opportunities for interacting with input signals. And again, an important component of this work is the novel innovation in developing anti-repressors, which I'll show how we can use this in the next couple of slides. So the next goal was to network transcription factors and direct them. So we can direct transcription factors in much the same way that you direct electric current. And this required that we iterate from protein engineering to genetic engineering. And so in this, we can direct transcription factors in series or in parallel or in a hybrid fashion called series parallel. So here's an exemplar of how you can create a multiple input, single output unit operation from 2 single input, single output unit operations, namely by pairing 2 non-synonymous transcription factors, in this case, 2 buffer gates that are network capable in their DNA binding function. We can create an AND gate of the variety that you see to the right. And the truth table illustrates the AND function perfectly in that 0 input corresponds to no output, and only in the presence of both the gray and the purple input do you turn the system on. Now, here again, we used green fluorescent protein as a proxy for the output, but you can use any output, including a non-coding RNA. So this allows us to couple this technology to other technologies like CRISPR. Likewise, we can construct network-capable anti-repressors in much the same way. And this results in a NOR gate. Now, the NOR gate is complementary or antithetical to the AND gate in that 0 input corresponds to 1 output. And in any other condition where you supply an input, the system is off. Now, NOR is actually very important from a programming perspective, and that NOR layering allows us to write any program of our choosing. However, we have a special capability of compressing circuits, and so this results in a novel programming language that's Turing complete. The way we tested that was by creating all 16 logical operations using this particular edifice. But that's only a small fraction of what transcriptional programming is capable of. To create other systems, or to date, we've created more than 1 million programs based on transcriptional programming structures, including things like digital to analog converters. And in this case, this particular device is useful in biomanufacturing in that it allows us to have maintenance of a set point, or we can actually use it in biomedical applications to control payloads. In addition to DAC, we've created bandpass devices that use repeated addition to control the on and off states. So in this case, what we can do is we can start with the system in the off state, supply an input to turn it on, and then at some time point later, we can supply a second input to turn it off. Now, the advantage of these systems is that they're much, much faster than the conventional way of turning a system on and off and a transition. Likewise, we can create the complement, and that is the band stop device, where we start in the on state, and through an addition, we turn the system off, and then at some time point later, we can turn it back on. So everything that I've described here has been built in a single chassis cell, or in this case, a single organism or microorganism. We've recently expanded this to communities of bacteria, specifically a special variety that are represented in the gut. So now we can execute transcriptional programming at the community level. And this is an example of one of our platform technologies that we're using to develop living therapeutics. Specifically, this allows us to control the population dynamics of the systems and may, in fact, be used for a variety of applications, in addition, including the development of smart probiotics. So we have a number of projects that are available. And applying this, we're interested in recruiting people to develop additional intelligent microbe systems for living therapeutics beyond this set that you've seen on the screen. We also are recruiting in the area of biosecurity, specifically biocryptography tools under developed for next generation security systems. Also in the area of next generation biosynthesis and biomanufacturing. Now, this is a fascinating project that we have in collaboration with an industrial partner. And I'm proud to announce that we have 3 new projects available this year in the area of programming mammalian cells, next generation bioelectronics, and applying graph neural networks or machine learning toward the development of new design rules for allosteric communication. We have a number of openings available. If you like something, come talk to me. If there's something on the screen that doesn't quite fit your bill, come talk to me anyway. I might have something for you. And with that, I'd like to introduce the Wilson Lab. Stay tuned for the lab video.
Our lab is located in the Engineered Biosystems Building, or EBB, one of the newest buildings on campus. The labs in the EBB have an open floor plan that integrates the office and lab space. Here's a view of our benchtop workspace. Our lab is outfitted with standard molecular biology equipment, including thermocyclers, microwell plate readers, a gel electrophoresis station, and multiple incubator types. Aside from being a beautiful workspace with great views, the EBB has extensive equipment available, including core labs with advanced facilities like flow cytometry, real-time PCR, light sheet fluorescence microscopy, mass spectrometry, and the like. Our lab is a collaborative work environment where you can expect to be trained by fellow grad students and collaborate on papers. We're hiring enthusiastic and talented candidates from a broad range of fields. Dr. Wilson joined Georgia Tech in 2016 and has expertise built up over decades in protein and genetic engineering and molecular biology. He's available for consultation basically at any time, but he's not a taskmaster, so being self-motivated is important in this lab. Overall, the Wilson Research Lab is a great place to work if you're interested in pushing the boundaries of synthetic biology and learning protein and genetic engineering techniques. We hope to hear from you soon.